Hi, this is Mr. Wartsky here, and today is the first screencast of Invertebrates. We are now in the animal kingdom, Kingdom Animalia. All right, so let's begin. So, in order to truly understand animals, we need to have the basic introduction. So, they are eukaryotics, multicellular, and heterotrophic. That means we need to consume our carbon and get our source of electrons. We store carbohydrates as glycogen. We have no cell walls. Instead, we have cell junctions like tight junctions, desmosome, and gap junctions, and our cells are held together by collagen, which is a connective tissue. We have muscles and nervous tissue. We reproduce asexually, and we have a life cycle that may include larval stage, and we'll talk about that in a moment. And we have Hox genes, and those code for transcription factors, which can turn on a set of genes at the same time. Other organisms do not have Hox genes. We do. So, in order to truly understand the larval form, that's like the intermediate form between the egg and the adult. So, if we take a look at a butterfly, and all insects are similar, they have the egg, and then we have out of the egg hatches the larval form here. The larval form would be like a maggot in a fly, or maggots in fruit flies. All right, so there's the caterpillar is the maggot of the butterfly, I guess. And then the larval form will undergo some type of change, some metamorphosis. They will pupate into an adult. So as they go through the change, they're called a pupa. Now, if you think about it, we are kind of like, we have this. We have the egg, we're born, and as we're growing up, that's all the larva does is eat and grow. So that's what you do as a kid. Then you pupate, that's going through the change, which would be what? Yep, puberty. And then you become an adult. So when you pupate, you go through metamorphosis. We just don't do it in private. We do it for everyone to see. And we probably should throw you in the closet, let you change, and then come out as the beautiful adult that you could be. So we are going to talk about invertebrates first, and we are going to look at the following phyla, starting with Porifera, cnidarians, they might also be called selenerates or selenerata, platyhelminths, which are flatworms, nematoda, which are roundworms, and lydia, which are segmented worms, mollusks, arthropods, and echinoderms. Or as a friend of mine used to say, echinoderms, but no, it's I like echinoderms better. So for each phyla, we're going to find the following information from major classes to symmetry, body layers, major structures, feeding, digestion, respiration, circulation, excretion, nervous system, reproduction, locomotion, and how important they are. Now, on the AP exam and in the AP class, in the curriculum, you're expected to be able to compare all those major body systems and from all organisms. So we are going to focus our attention on comparison, and we're going to do that constantly. So we're going to have to define some terms. Symmetry is how your body is arranged. We have three types of symmetry: radial, bilateral, and asymmetrical. Radial symmetry, the body parts are arranged around a central axis. So they're usually circular, so there's no front, there's no back, no left, no right. They only have a top and a bottom. And there are only two phyla that have radial symmetry, which are Porifera and cnidarians. Bilateral is the body's ranged along a longitudinal axis. So if you cut the body in half, you have the same parts on both sides. So you have a right, you have a left, a top and a bottom, a front and a back. That's the ventral is front, dorsal is back. And pretty much all the other phyla are bilateral. Now there is one class in one phylum that is asymmetrical, that has no symmetry, and one of these days I might even let you in on the secret, or you can find it out yourself. There's only one class of organisms or animals that are asymmetrical, that have no symmetry, no matter what anyone else says. So we have radial symmetry here. This is a sea anemone. Yep. So it's central axis. Bilateral, you cut down the middle, same one on both sides there. And this right here, I'll give you a hint, is the asymmetrical animal. That's it right there. So body layers. There are two main groups of animals when we talk about body layers. They're diploblasts and triploblasts. Now, their name implies the number of body layers that they have. Diploblasts have two germ layers. Now, germ layers are the embryonic layers, and they will become different things. In a diploblast, the two germ layers are ectoderm and endoderm. Ecto is the outer layer, endo is the inner layer. The outer layer will become the nervous system and muscles and tissue. The inner layer will become the digestive system. Now, there's usually a gelatinous material that holds the two layers together, and diploblastic animals have radial symmetry. So, if I tell you that, then you should know that porifera, sponges, and cnidarians are diploblasts. They have two body layers. Triploblasts have three, and evolutionarily they are more advanced than diploblasts. You have the ectoderm, which is the outer layer, skin and nervous system. The endoderm, which is the inner layer, which said that was the digestive system. That's true here. And then you have the mesoderm, which is everything else. And that might sound something familiar to you because we just talked about plants. The protoderm was the outer, and that could be similar to the ectoderm. The vascular cambium was the inner, like the digestive system of the organisms, and everything else, like the ground meristem, is the mesoderm, which is everything in between. Now, there are three types of triploblasts, of course. We have acoelomates, pseudocoelomates, and coelomates. And they're based on the presence or absence of a coelom. Now, all triploblasts are bilaterally symmetrical. Did I say bilaterally? All triploblasts are, yeah, bilaterally symmetrical. I was right. Okay, good. Let's move on. What happened there? I have no idea. So we have diploblasts, which are the endoderm here, the ectoderm's out here, and there's a gluey type thing in between. Digestion, outer layer. In triploblasts, we have the outer layer, we have the mesoderm, and then we have the inner layer, which is the digestive system. Now, the digestive system is just basically a tube, right? Mouth, anus, one long tube. The three triploblasts are based on a body cavity called a coelom. Now, a coelom is a cavity which is lined with mesoderm. So in the acoelomates, which is right up here, there is no body space, none. All the layers are right on top of each other. In a pseudocoelom, there is a body space, but it is not lined with mesoderm. So it's a fake coelom. That's what pseudocoelom is here. We have the ectoderm, the endoderm, and then the ectoderm, mesoderm, sorry, and then the endoderm here, which is the digestive system. Now, in a coelomate, we have the space which is lined with mesoderm. So you see this mesoderm here and mesoderm here, and then there's a space in between. Right? That is your coelom. Outer layer, skin and nervous system, your mesoderm, muscle tissues, bones, your liver, respiratory system, circulatory system, all that sort of stuff. Then you have the endoderm here, which is your digestive system. We're going to talk later about how the triploblasts form. First, you have a blastula, which is a hollow ball of cells. And then part of the blastula, cells move in to the center. And as the cells move into the center, they form this tunnel, right? This is a tunnel here. This is your archenteron, or the primitive gut. The blastopore will either be the mouth or the anus, depending on the organism. And then that will be the endoderm. So that's, we go from one layer, which is all ecto, to now ecto and endo. So when we talk about triploblasts, there are two types of triploblasts. There are protostomes and deuterostomes. So there's two types of animals. We have diploblasts and triploblasts. There are three types of triploblasts. There's acoelomates, pseudocoelomates, and coelomates. There are two types of coelomates, protostomes and deuterostomes. Now, in protostomes, the mesoderm is formed from the schizocoelous process. Basically, it's formed from the ectoderm. So the middle layer is formed from the outer layer. And we have annelids, mollusks, and arthropods are all protostomes. The deuterostomes, the mesoderm is formed from a process known as enterocoelous, which is the mesoderm is formed from the endoderm. So not the end of the mesoderm is no longer formed from the ectoligin protostomes. The mesoderm is formed from the endoderm. Now, the only phyla of invertebrates that has this are echinoderms. Now, deuterostomes are more advanced than protostomes. So the echinoderms are the most advanced invertebrate because they are deuterostomes. So we have other differences between protostomes and deuterostomes. On the third division here, we have one, two, third division. See, in the protostomes, the cells will be in between the cells, whereas in one, two, three, the cells are lined up here. That's one difference. The second difference is this blastopore. In deuterostomes, this will become the anus, and in protostomes, that will become the mouth. That's what proto means, actually, mouth first, and deuterostome means mouth second. Here we have the mesoderm forming from the endoderm, and here the mesoderm forms from the endoderm. So those are three differences between protostomes and deuterostomes. I might ask that question. So get them right. So the third division, how they line up. The second is which forms first, the mouth or the anus. And the third is where the mesoderm forms, from the ectoderm or from the endoderm. So let's talk about body systems. Digestion is how an organism breaks down the food. In diploblasts and platyhelminthes, they have something known as a gastrovascular cavity, which means they have one opening and a two-way flow. So they have one mouth that goes into this giant sac. They have the digestive enzymes breaking it down, and then it comes back out through the mouth. Yum. The alimentary canal that has two openings with one-way flow. You have a mouth, a pharynx, which moistens the food and moves it along, goes to the esophagus, which moves it to a crop and gizzard. Now the crop stores, the gizzard grinds. Now in us, we don't have a crop and a gizzard because they're combined. Our stomach is both a crop and a gizzard together. It stores as well as grinds the food. Then you have the intestines, which absorbs the nutrients and prepares the waste for export, and then the anus, where it comes out. Now, that is the same for all animals, from nematodes all the way through mammals. We all have an alimentary canal. The gastrovascular cavity was important because it was the first digestive system. The alimentary canal proved to be just a better machine. So here's the GVC. We have the mouth slash anus because it's the same thing. They shove the food in here, broken down in here through the enzymes being dumped into the GVC, and then the waste comes out. Some of the food comes out too. And that's actually how the cells on the outer layer are fed, because individuals with a gastrovascular cavity are really sloppy eaters. Really sloppy eaters. In the alimentary canal, you have the mouth and the anus, and you have all that stuff in between. Right? So hopefully you got the list. Not, you could always go back to here and I'll give you the list of the structures. Respiration is how an organism obtains oxygen. Now, all organisms need to obtain oxygen to produce energy aerobically, in which the oxygen is going to act as the final electron acceptor in the electron transport chain. It's the only reason you need to breathe. Now, there are different ways that oxygen can get into cells. One is by diffusion. Cells get oxygen by diffusion. Now, all organisms from porifera through annelids get oxygen in this way. There are no specialized structures in porifera, cnidarians, nematodes, platyhelminthes, or annelids. Gills are a centrally located area which contains a lot of surface area to volume ratio. Water washes over the gills, and the oxygen from the water will enter the gills in the blood by diffusion. Mollusca, arthropods, and echinoderms all have gills. Book lungs are similar to gills, but they're found in arachnids, right? They're internal gills, and tracheal tubes and spiracles are actually tubes that go from the outside to the cells in the inside, and the spiracles are the holes that the oxygen will enter. I hate to rush, but we're going to have to be done today. I'll pick this up next time. Hope this helped. Have a good night.